Good morning to everyone. Today I'm going to present the paper A Neural Correlate of Sensory Consciousness in a Corvid Bird, published by Nieder et al. In 2020. Birds' brain have organizational principles reminiscent of the mammalian brain, but lacks a layered cerebral cortex. Despite this, birds' behavior suggests conscious experiences. In this paper, they hypothesize that conscious experience originates from the activity of the NCL, the Nidopallium caudolaterale, and use a corresponding experimental protocol in which only the crow's internal state and not the physical stimulus properties determine the subjective experience. So the aim of the paper was to demonstrate the neural foundation that allows sensory consciousness exists in birds. To do so, they trained two carrion crows to report the presence or absence of visual stimuli around the perceptual threshold in a rule-based delayed detection task. As you can see in picture 1, the task was structured in this way. After the crow initiated a trial in the go period, a brief visual stimulus of variable intensity appeared in the 50% of the trials. After a delay period, a rule cue informed the crow how to respond if it had seen or had not seen the stimulus. In the stimulus trials, a red cue required a response for stimulus detection, whereas a blue cue provided a response for stimulus detection. In no-stimulus trials, rule response contingencies were inverted. Thus, the crow could not prepare motor responses prior to the rule cues, which enabled the investigation of neural activity related to subjective sensory experience. Trials were classified into supra-threshold, near-threshold, and no-stimulus categories. The crow's responses were classified into hit, correct rejection, miss, and false alarm. While the crows performed the task, they recorded single-cell activity in the NCL. They first identified 262 task-selective neurons that showed differences in firing rates for suprathreshold versus no-stimulus trials. The selective time intervals of these neurons that together bridged the total trial period were classified into stimulus-related and delay-related. Next, they compared the discharges during the crow's yes versus no responses in the 5 different trial categories, for both stimulus-related and delay-related neurons. As you can see from picture 2, during stimulus presentation, neurons responded mainly to stimulus intensity, because the responses during false alarms are similar to correct rejections in the no-stimulus condition. During the subsequent delay period, however, many neurons responded according to the crow's percept rather than the stimulus intensity. To find out whether the activity of the neurons was related to the crow's report for the same near-threshold stimuli, they compared the firing rates in the neurons' respective selectivity intervals for different trial outcomes. To do so, they used receiver operating characteristic analysis, deriving the area under the ROC curve as the probability of predicting the subjective yes or no responses for identical stimuli for the stimulus and the delay phases separately. They found out that during stimulus presentation, the neurons signaled the crow's subsequent report only mildly, but the activity changed to a predominantly report-driven representation during the delay. In fact, as you can see from figure 3, in the stimulus period, the choice probability for correct rejections and false alarms during no-stimulus trials is comparable to chance, but during the delay period, the choice probability for no-stimulus correct rejections and false alarms is higher than chance. Then, to explore the time course of choice prediction from stimulus onset to delay offset irrespective of neuronal selectivity, they applied a time-resolved population analysis based on the activity of all NCL neurons. They first trained a subtor vector machine classifier to discriminate yes versus no responses on the basis of the spiking activity. After training, the classifier was tested with new data from the same neuronal population, but for suprathreshold hits versus correct rejections in the absence of stimuli. Indeed, the classifier was able to correctly assign the new trials into yes versus no responses with particularly high accuracy at stimulus offset and toward the end of the delay, as you can see from figure 4. This indicates that a population of neurons contained information about the crowd's subjective experience throughout the trial. In conclusion, the results constitute an empirical marker of avian sensory consciousness. In particular, the initial sweep of activity is also mainly involved in unconscious vision, whereas activity correlated with consciousness is delayed relative to stimulus onset activity. maybe because only sensory activity that is strong enough can access awareness, causing a particular state in higher brain centers such as prefrontal cortex. In crows, the NCL may very well constitute the alien brain site of the process that leads either to high degree of activation, causing and maintaining information about conscious experience. These findings also provide evidence for the phylogenetic origins of consciousness, excluding that only primates or other mammals possessing a layered cerebral cortex are endowed with sensory consciousness. Consciousness in birds could either emerge 320 million years ago, when the last common ancestor between birds and mammals disappeared, or it emerged independently on the basis of convergent evolution on different branches of the vertebrate tree of life. Thank you for your attention.